For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Season 4, Episode 41 of the podcast, still known as Surfing the NASH Tsunami. Our review of the FDA's 2-day NIT workshop Monday and Tuesday of this week starts now.
This week on Surfing the NASH Tsunami.
Over the past decade, we learned so much about the limitations of histology, all the issues with screen failures, the astronomical cost of clinical trials using the histologic pathway. So I think for us, it's time for a change. The FDA feels like they learned so much about the current pathway and its limitations. We are talking about conditional approval today, and to qualify a biomarker for full approval, that will take time. But as you heard probably in the last session, that most experts in the field, they feel that we are ready to make that change for conditional approval today based on combination NITs.
There was a general agreement that first, inflammation is a driver and liver fibrosis is a killer, and steatosis is a marker.
These kinds of discussions, will just take an awful lot of additional discussions and in different settings to really drill down to the bottom of it. It takes a lot of infrastructure and collaboration to get there. Just even a little bit more of an alignment will have a huge impact. Maybe not today, maybe not immediately, but you start aligning a little bit and suddenly you have a path forward.
Every week, a global community of fatty liver disease stakeholders comes together to explore the most important challenges in diagnosing, treating, and developing medications for patients with fatty liver diseases. Join hepatology researcher and key opinion leader Professor Jarn Schottenberg, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and their guests, hepatology researchers, and key opinion leaders Naim El-Khoury and Laurent Castera, and Liver Forum Executive Director Veronica Miller, as they review highlights from this week's FDA workshop on NITs, on Surfing the NASH Tsunami.
Hi, this is Roger Green, host of the podcast, still known as Surfing the NASH Tsunami. This week, we had originally planned to record a podcast Tuesday afternoon discussing the first part of the FDA workshop on NITs, with plans to record material on the Tuesday afternoon portion later in the week and share it with you sometime in October. However, as the podcast progressed, we decided that the last afternoon was so important, it would be a better idea to wait until the end of the workshop and then record interviews with some of the key participants. So that's what we have for you today. We recorded 3 interviews on Wednesday, one with Naim El-Khoury, who was an expert commentator, panelist at the Tuesday's penultimate session, and then 2 of the folks who were part of the final session, Laurent Castera, who was one of the presenters, and Veronica Miller, who moderated the panel. They present 3 different, although somewhat complementary, perspectives on the science of NITs and likely impact of the workshop. The entire Surfing NASH team hopes you enjoy it. So first, we have Naim El-Khoury. Naim, how are you this afternoon?
I'm very good. How are you guys? Thanks for having me.
Well, thank you for joining. Fascinating meeting yesterday, hey? Last 2 days, hey?
Yeah, I think it's a very important meeting. I'm so happy that the FDA put on this workshop. I think, they had excellent speakers, and I think the discussions were very insightful and highlighted some of the issues that we deal with on a daily basis, whether you're in clinic or you have a clinical research site or if you're a drug developer. So I thought it was well received, and I think the number of attendees speaks to that.
There's a real thirst on this issue. The idea that they had 2,000 people on the webcast is pretty compelling, isn't it?
Absolutely.
What do you think worked best about it?
They selected excellent speakers. They covered most of the issues that we deal with in terms of designing successful clinical trials and getting drugs to patients that will make an impact in terms of outcomes and the natural history of the disease. I mean, anything that's organized by the FDA is going to get more attention because, they are the ones that will give us a pathway to drug approval. And for me and many of my colleagues, experts, we are trying so hard to develop an alternative pathway to what is currently accepted by the FDA, which relies on histology, repeat liver biopsies, NASH resolution, fibrosis regression. I think over the past decade, we learned so much about the limitations of histology, all the issues with screen failures, the astronomical cost of clinical trials using the histologic pathway. So I think for us, it's time for a change. I think the FDA feels like they learned so much about the current pathway and its limitations. They're very hesitant to provide an alternative pathway that will have different different limitations. So I totally understand where they're coming from. But to be honest with you, Roger, over the last few years, our confidence in histology has went way down. And I like all the artificial intelligence, new technologies, but at the end of the day, we really need to have an alternative. For me, for most of my colleagues, no one is asking to eliminate biopsy today. I think biopsy is still working for certain drug developers. We've seen very promising results with Madrigal based on histology and Akiro 89 Bio, Inventiva. So I think it's still a viable pathway, but I think we need an alternative one. And at the end of the day, I think our biggest ask is to provide us something for conditional approval. I think this is part of the confusion sometimes with the discussion that people are asking all these things from NITs in terms of predicting prognosis and what magnitude change will actually predict outcomes and things like that. But at the end of the day, we are talking about conditional approval today, and to qualify a biomarker for full approval, that will take time. But as you heard probably in the last session, that most experts in the field, they feel that we are ready to make that change for conditional approval today based on combination NITs.
Well said, Naeem. I am aligned with most things you said. Also from the start, I think the recognition the field gets from having FDA host such a workshop with great speakers is just tremendous. I think it'll be very important for the field moving forward. And coming back full circle to what you said in the end, I think the discussion, the way I understood it, went that way, that we're saying if you accept an imperfect surrogate for conditional approval, we're ready to substitute that with another, maybe not perfect yet, non-invasive surrogate, and do not get full approval, but we'll keep these people on drug. We'll come up with a full benefit after a certain time. And I think that's the way forward, what I heard. And I think that's what you just said. So that's very encouraging. I'm not sure they're following that. I have one more thought I want to share, but I'll hand over to Roger first to hear what Louise and you say.
I obviously agree with what Naeem was saying. And I thought when I watched the sessions, Naeem, you were the first person to bring passion. Not that the other people didn't bring data, statistics, and the normal way that we do it about it's all about the data, it's all about the outcomes, and let's be clinical here. But when you started to discuss in your session, you brought the real impact to patients about the ballooning hepatocyte, the screen failure rate, the way this affects people's care who are just as affected by the disease but can't be used in a clinical trial and therefore miss out. And it was echoed in the final session, but we do need to send that message to the FDA. And I think the one thing I'm taking from this is the FDA think this is an important enough issue to convene a 2-day conference to be able to listen to you guys in a concentrated period where it's all focused. That does say an awful lot as to where the field is heading, and the data that came out was priceless, and that discussion, but the passion that you came with was a swinger for me, but I swing from that passion hymn sheet.
Well, thank you, Louise. We're all passionate about this. We may have different styles of presenting our ideas, but I think, as you said, it came across really well with the last session. I think there was consensus. As you mentioned, these are real issues that we deal with in any research site on a daily basis. I brought up an example of a patient with F3 fibrosis that has, activity score of 4 points, and they have no ballooning, and now they are a screen failure. And what disease entity we're dealing with? This is not MASH. This is not the aggressive form of the disease. Obviously, it is when you have F3 fibrosis. How can we deny this patient participation in a trial? So I think that was one issue. The other issue we talked about is we all agree that disease activity based on the NAFLD or MASLD activity score, based on steatosis, inflammation, ballooning, this is the driver behind fibrosis, and fibrosis is the driver behind outcomes. So disease activity, very important. But the point we tried to make is the definition of NASH resolution may not actually reflect improvement in disease activity, and the patient can actually have significant improvement in disease activity going from a NAS of 8 to 2 and still have 1 point on ballooning, and they will qualify as a non-responder in a trial. So you could be missing out on drugs that might be doing very good things in the liver based on the definition of NASH resolution, and the reliance on ballooning to diagnose NASH and then the requirement to lose ballooning to call it NASH resolution with that Brunt et al. Paper that that looked at the consensus between expert pathologists. I mean, that was an eye-opener for me. If you guys recall, it was 9 expert pathologists, like top of the line, and they had to review 10 slides and score ballooning 0, 1, or 2. 0 meant that they had 0 to 5 balloon cells, 1 was 5 to 50, and then 2 was more than 50. And on the same slide, 2 expert pathologists can score 0, meaning less than 5, and another one will score 2, meaning more than 50 cells. If we're putting that emphasis on NASH resolution and we have these major issues with ballooning, that should be an eye-opener for anyone who's trying to develop drugs and for the FDA. The other point we made very clearly is that, we don't believe today that there's an NIT by itself that can become reasonably likely surrogate endpoint. But I think a combination of NITs is there today. And one thing we're talking about with NASH resolution, again, let's take it a step back and say, what are we determining with NASH? We're looking at steatosis, disease activity with inflammation ballooning, and then we have the fibrosis piece. And we have NITs that can look at each of these pieces in a way that's more reproducible than a pathologist. So that's what we try to argue at the end, that if you have combination NITs, especially if you have 3 of them showing you improvement, that this should carry the same weight as NASH resolution.
So something, Naeem, and I wasn't able to follow all aspects that wasn't discussed intensively, is that certain markers could be used to reflect target engagement. And I think that might be important. There was, I think Arun mentioned that liver fat doesn't really predict the outcome, but if you have a drug that reduces liver fat, of course, you want to have that in a treatment response biomarker to show target engagement and effect. And there are some drugs we're developing that have new measures, for example, the sex hormone binding globally, that's been altered by resmetirom. So there wasn't an intense discussion on that, I believe, but what's your take on how we could combine that to amend those surrogates?
No, I completely agree. I think the mechanism of action should be taken into account if you're going to develop alternative pathways. If you have a metabolic drug that's known to reduce liver fat, the way I would think about it, with combination NITs, that I would like to see probably 3 unrelated NITs go in the same direction, and I would design it in terms of MRI-PDFF reduction by 30% or more, plus reduction in ALT by 17 units or more, plus improvement in one fibrosis biomarker, and that could be potentially reduction in liver stiffness on FibroScan. The consensus was about 30% reduction from baseline, or maybe a reduction in the ELF score, and then looking potentially at a combination of these in a test. For example, you can do the MAST score. So I think this is the way I would think about it for a metabolic drug, that you think you're going to target upstream, decrease liver fat, that will lead to downstream improvement in NASH and fibrosis. If I have a purely fibrotic drug that I don't anticipate to improve steatosis, probably I'm going to look into 2 unrelated fibrosis biomarkers. So now I want to see improvement in potentially liver stiffness on FibroScan and ELF, or maybe ProC3 as a more dynamic fibrosis biomarker. But again, I want to see that consensus, and I want to see the biomarkers moving in the right direction. So I think there are ways to do this. I think, again, for a metabolic drug, let's say something like a THR beta, in my mind, if you reduce liver fat, ALT, and liver stiffness, that should carry the same weight as NASH resolution. Okay, and if we're going to accept NASH resolution, we should provide that, alternative pathway. Fibrosis regression on biopsy is still probably a higher standard for me. But again, since we are providing the NASH resolution as an accepted endpoint, I think this combination NIT approach is reasonable. I do like the combination biomarkers, and maybe I'm biased here, but my favorite is MAST. And I think, one key thing is people don't understand this about these biomarkers, that by having the MRE and PDFF, some patients can actually reduce liver fat, but their liver stiffness will go up, right? We see this as they progress toward cryptogenic cirrhosis. So having that MRE piece and just making sure that it's not going the wrong direction, even if you cannot make it go down, because we've struggled with MRE in the context of short-term clinical trials, I think that's still very meaningful with MRE specifically, because at least you can avoid that worsening piece. And that, if the MRE is going up, it's going to affect that score, and it has, high weight in the MAST score. And obviously there are other ways to do this, but, I would say probably MR technology is superior to FibroScan technology, especially in terms of quantifying liver fat. I don't think anyone will question that.
Let me step back a little bit. First of all, let me say that I agree with everything all you guys have said so far in terms of how the world works. Okay. But the thing I have in the back of my head is the way a social psychologist wins a Nobel Prize in economics is by proving that we're not averse to risk, we're averse to loss. That people will take tremendous risks to avoid losing things. And then the question becomes, how do you define loss and how do you define big loss? We define loss in all the ways you've been talking about. But part of the reality of government and bureaucracy is they define it very differently. One of the conversations we've had today is with Veronica Miller, who obviously lives close to this from a slightly different perspective. One of the things that had struck me was they spent the first session rehashing Aduhelm and Makena and rosiglitazone, right? So they want to explain why they don't get things wrong, because when they do get things wrong and there's public reaction, it's catastrophic. So my suspicion is that we're going to need to prove a tougher case to them than we would have to prove to ourselves or the other academic colleagues. And in that regard, I guess a couple of things that came up yesterday, I think. First of all, one is this question of if the path to regression is not the same as the path to progression, then what does that say about NITs? And more generally, the more you ask the agency, or really any regulatory body, to consider different solutions to different questions depending upon the specifics of the drugs, well, we live in that space, right? But they don't. So I'm a little concerned that if we make it too complicated, the innate fear of making a change and getting it wrong will dominate. What do we do? If the goal was to make the first step simple, what would we lean on?
No, I mean, I totally understand the concerns. I think, what they realize is whatever pathway we've had, it was a breakthrough. And I really commend them on actually providing us to a drug approval pathway. I think this is what started this whole NASH drug development field, and that's why we're here today. So I think what they did in 2013 was really a big step, and probably they didn't have all the information they needed at the time, but they felt that they needed to act. But we've learned so much about the impact of this pathway on patients and drug development today. So I think it is time to change. Are we in a place to say we know everything we need to know? No, I don't think so. I think there's a lot that we need to learn. But I'll tell you my math, Roger. In our clinic, so we're a hepatology clinic. So by the time the patient is coming to us, this is great because at least they know they have a liver problem. But I see 100 patients with NASH, and only 5 of them will potentially screen for clinical trials, right? So only 5 out of 100, this is in a hepatology clinic. And then when we do all the inclusion-exclusion criteria biopsy, only 1 or 2 end up actually enrolling in a trial. So when you take this to the general population, I mean, this would be probably 1 in 1,000 patients with fatty liver. And these are not the statistics that we have shown over and over. If you remember in our, prospective study, myself and Stephen, we showed that the general population, 14% have NASH and 6% have significant fibrosis. With the current pathway, we turned the most common form of chronic liver disease into a unicorn disease that only exists in, 1 in 1,000 individuals. And just this is not the case. So I just don't want to be in a situation where it's like paralysis by analysis when you have actually a bad pathway that's really not reflective of the patient population that we're trying to target. Every surrogate has issues. Everything can be affected by potentially other disease states. And that's why we're insisting on combination NITs.
I share some of the skepticism Roger mentioned, and however, I do see how the FDA wanted to help the patients and the field to move this forward. So the one comment I thought about for a little bit was the FDA statistician's name. Not sure you realized her. She in the end said, well, just enroll 100 million people and do a full outcome trial. And it sounds like a very straightforward shot, but the problem of, the way trials are currently run, that's impossible. And it negates a lot of the troubles we're going through. So I wasn't sure how to place that. How did that sound to you?
Yeah, I mean, in cirrhosis, we are at that point now, and we are, able to enroll patients potentially without a biopsy. I think we have confidence now in NITs to diagnose cirrhosis. But as you said, once you have F2 and F3, I mean, what are the outcomes? Are we talking about decompensation, ascites, HCC? I mean, that will take a very, very long time, right? And we've seen this even in cirrhosis trials. If you're not including patients with portal hypertension, the event rate is about 3, maybe if you're lucky, 5% per year. So let alone taking patients with F2. And then to do it without biopsy and again, enroll 10,000 people and wait and see how many will develop cirrhosis is just not feasible at this point. So I agree again, in a perfect world where cost is not an issue, sure. The the MRIs we do are not also cheap, so that's another thing to factor in. Yeah, so I don't think that's realistic. I mean, it'll be nice to do, but I don't think it's feasible or possible.
Naeem, one of the most compelling things that was shown yesterday, although it wasn't covered particularly, but something we all know, is that it isn't until you get to F4 that liver-related mortality becomes a leading cause of death. If F0 through F3, you have significant impact on mortality, but it's not necessarily liver mortality per se, which to me, by the way, is the best argument not to do your million-person 10-year trial. If you had enough money to do that trial, how many people would die of coronary diseases and other metabolic-related diseases while you were waiting to see if they develop cirrhosis or not?
Yeah, and to be honest with you, Roger, we are so focused on liver-related outcomes and hepatic efficacy endpoints, but we need to also potentially get beyond this and think about cardiovascular outcomes, metabolic syndrome, improvement in each component. Obviously, we look at overall mortality, but there are ways to actually think outside the box and come up with different pathways. But at this point, the FDA made it very clear that to approve a NASH drug, it has to show hepatic efficacy. But again, this is probably not the right way to think about this systemic disease. I think we need to potentially go back to the drawing board and figure out a different way to design trials focusing on overall mortality, cardiovascular, liver, chronic kidney disease, progression to type 2 diabetes and non-diabetics, progression to CKD in people without CKD at baseline. These are all meaningful outcomes.
Well, maybe there's the trial that needs to be written that you've just described. We need a prospective study using non-invasive technologies for metabolic health that include a large number of these ones that we've discussed for the last couple of days, and use lifestyle rather than anything else, and look at what that longitudinal study does. Now, we see lots of longitudinal studies like that in vitamins. They did a 10-year study with 10,000 people on multivitamins, 10,000 not. And ultimately, all it did was deliver what we suspected it was going to deliver, expensive poo from one group versus the same health metrics in another. And we've not seen that longitudinal study that looks at the effect of poor liver health and measure it in mental health, because we know that one of the biggest issues with metabolic liver disease is this depression, clouded thinking. People just don't have the energy. And we know that when we defat the liver, mental health is something that improves quickly. So maybe by missing all of those parameters in the past, we've not done ourselves justice, but we're not looking at drugs for that. We're actually looking at monitoring the liver's impact on the body as a whole. Maybe that's what we need to go back to, looking at what happens when the liver either improves or deteriorates. Roger is right. The FDA, like any psychology, you are affected by your future decisions by the results of your past. And it's very difficult to get beyond that. And I would rather go into the future making the wrong decisions for the right reasons, evidence-based, than actually make the wrong decision for the right reasons and use a surrogate marker that has a poor outcome and a risk of harming individuals. And we have seen lots of drugs go forward that were put there for the wrong reasons, even with good evidence. So I think at the time, so there is a balancing act, but psychology is we don't take that step forward because we're affected by our past experiences. But this meeting shows that maybe they will, and at least consider it.
I love this, Louise. I'm going to start using this. So making the wrong decision for the right reason.
Making the right decision with some of the wrong reasons, because they would say using NITs is not the right one. Whereas we've made some drugs that were approved, right drugs, but with the wrong decisions behind them. I think have I clarified that better? Right decisions, wrong reasons. Wrong reasons, right decision. Yes.
Depends on your risk affinity, right? And obviously the side of the table you sit on. But in generally, I agree.
If the agency is looking to take next steps, and the next steps certainly aren't going to be as big as everything that we know, where do you, is your sense of, based on what you heard yesterday, where do you think the next steps are most likely to come?
There's a lot of data that's being generated as part of the phase 3 clinical trials with a lot of NIT data. These patients are being followed long term. The FDA will have also more data to look at in the near future. So that's good news. We're going to also have a better understanding about the risk of outcomes in these clinical trials and how many will progress to cirrhosis. So we can also start thinking in a more informed way about the sample size if we want want to do outcomes trials. I think the prognostic data with the biomarkers is becoming stronger every day, especially for the fibrosis biomarkers. And I think the combination NIT field is also maturing, and we are having a better understanding about what it means to have improvement in PDFF and ALT, for example. And we've seen the odds ratios or the hazard ratios with the combination being better than each test alone. I cannot wait to see more with things like MAST and MeFib, and looking even at fast changes, and also changing the categories of these NITs going from high risk to medium or low risk. So I think a lot of data is being generated now, which is good for the field. The way I would love to see it evolving is to have an admission that, hey, what we have today has caused so many problems and is really not a great pathway, that it is time to provide an alternative pathway. And I would like to maintain the status quo, but just have another pathway based on NITs. That would be a dream come true for me, and I know for many of my friends like Jorn and other experts in drug development. So again, to reiterate, all we're asking for, do not get rid of liver biopsy today. I think it's still a viable pathway for some drugs. Just give us one alternative pathway based on combination NITs and taking into account the mechanism of action of the drug. And this will be pre-specified a priori to the trial, obviously. And I think we can, by doing this, decrease screen failure rates, be more inclusive in clinical trials, generate data from trials that will actually be similar to what we see in the real world. And I'm pretty confident that will actually help our patients long term. And at the end of the day, we share the same concerns with the FDA. I mean, I don't want to have drugs that don't work for outcomes at the end of the day. I don't want to medicalize everyone with fatty liver disease and say you need to be on pharmacologic treatment. These are the last things we want to do as physicians. We just want to have a viable pathway that will help these patients, with the end insight is that you need to show improvement in outcomes.
Can I ask a question? Okay, in my real world, in the future, I suppose my fantasy world, say the FDA go to approve non-invasive surrogate markers. Does that open the way to add those non-invasive surrogate markers to other trials? We can't do a cardiac trial and use liver biopsy to look at the effect of liver health and the underlying liver disease in those populations. We can use VCTE by FibroScan and things like that to look at, because at the moment we don't really know, as far as I can tell, in most of these other drug studies that use the liver to regulate their mechanisms, either upregulate, downregulate. They don't know their underlying fibrosis scores. They don't know it as well. We know that 60% of patients may have normal liver function, and we use liver function 5 times the upper limit of normal. These sort of ranges. By taking that leap of faith, going non-invasive, do we strengthen the ability of all other trials to be able to use a non-invasive liver marker to get better outcomes and detail their populations?
Yeah, I mean, I think to be clear, what you're referring to is that even potentially treating patients with NAFLD or MASLD and defatting the liver, that may lead to better outcomes in terms of cardiovascular outcomes and maybe patient-reported outcomes and quality of life. And that's a big unanswered question, to be honest with you. And today, I think we can use MRI PDFF to look at the effects of an intervention of, defatting the liver. And it would be a very large study if you want to look at cardiovascular outcomes and overall mortality, patient-reported outcomes. But yeah, of course, if we have a pathway for approval in terms of liver efficacy, that will lend more credit to these NITs. But I think what you're trying to say is that fatty liver, period, is not good for anyone, and it can lead to other healthcare issues. And what if we treat fatty liver? I think that's a very big question that needs to be answered, because there's so many confounders, right? I mean, most of these patients have obesity, obstructive sleep apnea, diabetes. We don't know what's affecting their quality of life. Can you tease out that the liver by itself is the driver behind some of these? A very big question to answer. I really can't tell you if I know a good scientific answer to this question, but I like where you're going with this.
So let me put on my commercial hat and then my statistician hat, maybe my statistician hat first, right? Which is that I don't think the question winds up being whether the liver is solely responsible approval, but it's whether if you ran some kind of a multivariate analysis, would you find a dimension on which the liver dominated and would have sufficient p-values to be significant and sufficient impact to care, number one. Louise, I think the interesting thing about your question is that no commercial drug developer is going to ask any bigger than the narrowest question they need to ask to get approved. Now, actually, we're a little spoiled in fatty liver because people have understood that they needed to take a look at NITs and stuff like that, because if we don't move the science forward, even the approvals are going to be overly limited. So we see more science in these trials than I have in other places that I've worked, other disease categories that I advised earlier in my career. But commercially, you wouldn't want to have your heart failure drug, your preserved ejection fraction drug, meet its endpoints and get nailed on a safety problem because you did a liver test.
Yeah, but I'd rather have my drug doing my ejection fraction knowing whether the patients who failed had fibrosis or cirrhosis and didn't just assume it was my drug failing.
That's a post-approval trial, and normally you'd look at that going for a secondary indication. If I'm responsible to shareholders, the first thing I have to do is get to market. And it's a little bit like writing a doctoral dissertation. You make the question really narrow because you want to make sure you can anticipate all the incoming. I think this in that regard is similar. That said, I agree with you totally that that's a study that should get done. You know, if 50 years ago it would have been in Framingham, right? It's probably a UK biobank now if you play it right.
Possible, yeah. I do have to go, guys, but I just wanted to thank you again for having me, and I wanted to thank the FDA also for putting on this amazing workshop. I really enjoyed the interactions and listening to my colleagues and the FDA perspective, and I think we're in a very good place, and we're hoping 2024 would be even a better year for our patients with NASH or MASH, and we did it despite the difficult path to approval. I think, we've done it with several, compounds now showing very promising results, and hopefully the FDA will grant us that first approval next year. So thank you again.
Well, Naeem, congratulations to you for being part of this event and playing such a powerful role in it, and we look forward to seeing you soon. And thanks for joining us today.
Yeah, see you later.
Second, Laurent Costera. And we have with us now our good friend Laurent Costera, who actually was with us last week. Laurent, 2 weeks in a row, huh?
Yes, absolutely. Good afternoon.
That's virtually a first for a non-host. So I think you go on the wall in the NASH Tsunami Hall of Fame as a 2 week in a row guest who was not a host on either week. Congratulations. We will be sending you your notification shortly. How are you doing today?
Fine, thank you. How are you doing?
Well, we have Louise with us, and she's just back from Australia, and we have Jorn with us, and he's on mobile phone. So lots of logistics, but everybody's here, and that's great. Why don't we just dive in? As one of the few non-Americans to present in yesterday's meeting, I'm wondering what your impression of the meeting was as somebody who really hasn't seen a lot of FDA before. So how did it strike you?
I think it was well organized and very comprehensive, addressing all the different aspects of NIT, of course, starting with liver biopsy, AI. Could we improve the limitation of scoring systems that are well known, such as ballooning, for instance, that is really a hurdle in trials, and then reviewing the different NITs, including imaging, circulating NITs, and finally trying to put this in perspective, how we could use in the future in the trial NIT, and can we get trials free of liver biopsy. My sense is that after this meeting, I'm not sure we're there yet, but to me, this is a strong signal to the community that FDA is considering the possible use of NIT. I think this is a very strong signal, and as you know, usually the world is following what FDA is saying and deciding. So this is my take.
If I can say something quickly, Laurent, also congratulations on a great presentation. I followed most of it. To some clinic aspects I couldn't follow all of it, but I agree with you that in that sense, this wasn't a typical scientific meeting we normally have to discuss data. This was a meeting with the regulators, and actually it will bring the field into attention of a much broader audience than we normally reach. It would be one of my expectations. I'm not sure, as one of the speakers, what your expectation there is, or if you heard something, because obviously FDA is an official government body and they make the rules in that context. So I think it was a great opportunity to be able to present there and share our thoughts. I felt all talks were given very deliberately and under that impression that this is not just a scientific meeting, but in general, I'm aligned with you. And I also heard heard the message that we want to move away, but maybe are not there yet. Maybe one or two speakers could have pushed the idea more to leave biopsy quickly, because at many levels I heard that it's so inaccurate and the concept of ballooning or resolution of steatohepatitis is really difficult. But again, we need the data is my bottom line. Not sure how you see that.
I think I concur with you. I mean, in a nutshell, I think there was a general agreement that first, inflammation is a driver and liver fibrosis is a killer and steatosis is the marker. So, of course, we need to take into account inflammation. This is important. And also with different drugs. I mean, you have drugs that are more active on fibrosis, and the metabolic drugs mainly act, let's say, on inflammation. So you cannot get rid of inflammation. I think, again, my sense was there was a general consensus that ballooning is not reliable. And this is the main drawback of the histological scoring system, whatever scoring you're using, the NASH CRN or the SAF. And this is related to the lack of reliability, the inter-observer viability, even among the best pathologists. And this is exemplified by the paper by Elizabeth Brunt, published last year, showing that even 9 expert pathologists do not agree on the number of balloon cells. And this is quite worrying because, I mean, we all know and all agree that using liver biopsy, if a drug comes to the market, will be a clear bottleneck. One of my take-home message was that the risk is to have a drug approved and having a lot of patients that would qualify or benefit, let's say, for treatment and will not qualify according to the criteria. And this is not what we want, of course. So this is my opinion and my perception that there was a general agreement that ballooning is really an issue and we should find a way, or AI might be an option. Not sure it's the grail, the holy grail, but clearly ballooning is an issue. Another issue is we are far from having the perfect NIT that is able to capture with enough granularity all the spectrum of the disease and NASH, especially. Fibrosis, yes, I think NIT, at least for diagnosis, are doing not bad. But NASH, I don't think among imaging or circulating biomarker, there's a single NIT that is able to capture NASH. I would be curious to get your opinion, actually. I don't want to be the only one talking.
I was going to make a comment first, then I'll ask Jorn and Louise. So when the episode that we ran on the Brunt paper, which was one of our NASH Tech episodes the year that was released and one of the most listened to episodes we've ever had, Quentin Anstey was on that episode, and afterwards we did a conversation with HistoIndex. The reason we did that was because the intermediary solution, or the shortest term solution, would simply be to redefine the ballooning variable, right? That one problem is ballooning might matter, but using a 2-point scale based on observer definition is a highly unreliable way statistically to bring that into the process. So the conversation was something like a HistoIndex Q ballooning score, where you could get much greater alignment about how much ballooning was present as a percentage or as some kind of a continuous variable as compared to simply a 2-level nominal variable might be a first step. Were you on that episode? I don't recall that you were a host yet, but you might have been on the episode.
Yeah, I remember that episode. I agree. I mean, that paper is really a landmark in showing us some of the shortcomings of the currently accepted surrogate that's reasonably likely to predict the outcome. And one of the arguments that's been brought forward, and Mazen also compared this to HCV eradication, where that was accepted, is if we're ready to accept such a difficult endpoint, why don't we just go the next step and then accept an NIT? And Laurent made some comments. I think I concur with him that in the realm of fibrosis biomarkers or assessing fibrosis regression, we are better situated today. When I recap the ELF data, I wasn't convinced that ELF is the perfect biomarker. Vlad Ratziou made the point that we should look at categories, which again brings us down to, maybe 2 or 3 categories. I think I saw some data that you presented, Laurent, unpublished data, so not peer-reviewed. I'm not sure how much you can share with us at this time, but the changes of VCTE in terms of percentage reduction, that looked pretty promising, and it was linked to outcomes. So this could be, even in a treatment response biomarker for fibrosis, one way forward.
Yes, so let's go to fibrosis. We are discussing about inflammation and NASH, But yes, I cannot comment further from what I've shown yesterday because, as I mentioned, these data are not published yet. And Vincent Wong from the University of Hong Kong graciously allowed me to present the data in support of my argument on the fact that we could use NIT, especially liver stiffness, using VCTE as a surrogate of response to treatment, at least in terms of improvement of fibrosis. So the reasoning was the following. There is growing evidence, whatever the technique you're using, let's say, is a VCTE, and of course, lower level of evidence for MRE, that if you have an increase over time in liver stiffness, so let's go back to VCTE, starting from, let's say, 15 to 20 kilopascal, not only baseline stiffness is of prognostic value, but the changes, the kinetics over time, if you have an increase, will be related to the occurrence of liver-related event, liver-related mortality, and overall mortality. Then you can discuss the magnitude of the decrease, whether it's 20, 25, 30%. There's no consensus. This is what I've shown. Now, the major, let's say, kind of philosophical issue is this, don't forget, these markers have been designed for progression of fibrosis, not for regression. And the question is whether if you observe regression, does it mean that the outcome are going to regress? And this we don't really know. One of the issues also, as I've shown yesterday, is the spontaneous variability of liver stiffness over a short period of time. And there is variability. And I hope after this meeting that I convince people that we should look at this data. People have this data. You just need to go back to database and look. There is variability. And the study I presented is a monocenter study from France, actually, retrospective in patients with chronic liver disease, mostly viral, not NAFLD. So we need more data. But there are also data, again, from Vincent Wong showing that in patients, these are NAFLD patients, with 2 liver stiffness measurement within 6 months before a liver biopsy, those with liver stiffness above 8 kilopascal, 1/3 go back to normal value. And this was ascertained by a liver biopsy with no fibrosis. So in other words, meaning their variability and their false positive. So this is important to take into account if you are to use liver stiffness as a surrogate of improvements. In other words, the decrease, does it mean that is it real improvement or is it just false positive? Now, the data I've shown are very important data because it's a large set of more than 10,000 patients worldwide, 16 centers that pool their data and showing that if you have a regression with a liver stiffness decrease of 30%, there was a significant decrease in liver-related events. So put in other words, if you have an increase of 30%, this was a short period, about less than 2 years, there's a 20% increase in liver-related events. If you have a decrease on the opposite, there's a 40% decrease of liver-related events. So this is a very important information. Now, is this related to some false positive? Why is it higher in terms of regression than in progression? This we don't know. And I don't want to go further because, again, these data are not published. But the data are there and they will be presented soon and also published, hopefully. But these are very important to move the field forward because this is what people are expecting.
I agree. And thank you for jumping with me from NASH resolution to fibrosis. I agree with you that I think the data that's been presented on fibrosis is just closer to acting as a true surrogate. And some of the data you detailed with us here, I think, shows that. So looking forward to see the full conference report or paper when they're coming out, of course.
So although, Laurent, you make a really interesting point. 2 years ago, in the discussions about use of NITs, people were making the distinction between NITs to qualify people for trials versus NITs to assess drug efficacy, specifically for the reason you give, which is that we were disqualifying people from trials who clearly had NASH because of the ballooning issue, but that it wasn't clear yet that the disease regresses the same way it progresses. How much progress do you feel we made on that issue yesterday or was made by the presentations yesterday?
The point you're making is important. We have seen in our last session with the Nimble and the Litmus how much data have been generated and especially meta-analysis. So for instance, there is this large meta-analysis analysis published in the Lancet Gastro by Litmus very recently, or more than 2,000 patients, showing that NITs, the most used NIT that are liver stiffness, FIB4, and ELF have prognostic value. I think this is important. So again, the amount of data has tremendously increased over the last 2 or 3 years and increased our confidence in how we can use NITs. Now there are several settings. Again, this is what I've shown in my talk. If you want to enrich population from primary care with risk factors for trials, I think we have the tools. And actually we have 4 guidelines that are currently aligned for that. So this is kind of standard of care. And this has been achieved over the last 2 years, starting with the EASL guidelines. So FIB4 as first test, then according to local availability, either VCT or ELF test. And then when you're in the liver clinic, of course, you want to distinguish who are the patients who would qualify for a trial. And again, we have composite score, for instance, Agile 3 that can improve slightly, maybe. We need more data. The confidence in advanced fibrosis, because there's a risk of false positive with VCT, as you know, in 20 to 30% of cases using the recommended cutoff. And then you may use composite scores such as FAST, or if you're lucky to have MAST or MEFIB with MRE, to be able to spot the patient of at-risk NASH. But still, it's not the grail, but it's helpful. And some of them are probably easier to use in practice, like FAST, because VCT is widely available. Maybe not as much as you would like in the US, but still worldwide it's widely available. And this can be implemented tomorrow. But we need more data and also independent and further validation, head-to-head comparison, and we're not there. And then there's treatment response. And treatment response, I think the shift in paradigm is very important, but we need to generate a lot of data. And still, I think we might need liver biopsy for that purpose.
I'd like to come back to something Quentin and I think some other people showed, is that the response of transaminases, which seems like the most known test and most widely available from drug treatment, particularly at the beginning, it's normally pretty sharp and then it kind of plateaus off. I was born because I do like ALT assessment, but it seems that it's an unbalanced response, particularly early when you put someone on drug, and then the long-term benefit is not as clear. So that's, of course, also of concern if you use it in a composite score, Laurent, and it might need a composite score that maybe doesn't have that more volatile ALT or AST in it. But I'm not sure here, so we'll have to see more of the long-term data. But something that was shown is, the sharp drop of the first 4 or 12 weeks people on drug in those liver enzymes. So I'm not sure that's a good predictor of treatment. It wasn't a predictor of treatment response.
You're absolutely right. The NIT that you use that include transaminase, FIB4, I don't think is a good marker for that. On the other end, transaminase is quite a robust marker of inflammation. If you normalize transaminase, it tells you something. The issue is what happened afterwards, and this you need another test to capture. Again, I think we can learn from viral hepatitis. You might remember that I've shown a study that was published about 5 years ago from China with an HBV patient treating with NUC and around 150 or 60 having per liver biopsy over 5 years. And there's clearly a 2-phase decline. I'm talking about liver stiffness. And there's a rapid decline that paralleled the normalization of transaminase within 6 months. And then afterwards, he has a much slower decline. So the first rapid decline is likely related to inflammation. But once you have normalized transaminase, liver stiffness still decreases at a slower pace, and this is likely related to fibrosis. So again, we don't have many studies, especially in Hep C, but at least in Hep B, I think we can learn from this kind of study, and would not be surprised that it would be the same in MASLD or MASH.
That's true, and that's biological plausible. So that's something that also was said multiple times yesterday. So it will fit the concept of how that biomarker should behave.
Louise, do you have a question for Laurent?
No, I think he was covering it beautifully there. It was a great sum up and some great data presented. So encouraging, shall we say, even more so moving forward.
I suspect that if that data, the unpublished data, is presented at AASLD, it will have a profound impact. I think you're right. That's that's just a point that nobody's brought any kind of conclusory data to yet. That was pretty powerful stuff, I think. I couldn't agree more.
Actually, I did have one question, Laurent. When you were talking about the 8 kilopascal level, where people change their kilopascals from 8 and drop, we often see in clinical practice when we ask people to make a lifestyle change during that period, they make a lifestyle change, their kilopascals and their steatosis levels drop. Are they false positives, or are they rapidly changing positive to negative because they've had that intervention of a FibroScan, do you think?
I'm not sure I have the answer to your question. That's a good point. I have the same experience. And the issue is BMI is a major confounder of liver stiffness. So there might be some inflammation, steatosis, that for sure. I don't recall whether there were CAP values in this study as well, documenting, the decrease in CAP value, because this also we see when people go on a diet. And I think it's a good incentive in practice, actually showing the numbers, because this talks to our patients. But the question is whether it's the decrease in BMI that is significant, that is followed by a decrease in liver stiffness. But this might go from, let's say, 8.5 or 9 kilopascal to 7 kilopascal. So how significant is that? But this is something that we have seen as well. Unfortunately, again, there's just a single study documenting that. And again, I would be interested and curious to see other study, whether this is reproducible, and if other investigators, this study comes from Asia, from the group of Vincent Wong, as I mentioned. But is it true in Europe? Is it true in Australia? Is it true in the US? We don't really know. So again, would be interesting to see more data on that issue. But again, this is critical, because if we're going to use this as a surrogate, you want to be sure that retest works, repeatability. This is something that needs to be addressed, I think.
And I think you said in the meeting that you have to remember the method of action of the drug and steatosis. Some of these drugs target steatosis rather than fibrosis. Was that one of the outputs that you came up with in the
yes.
round table?
Just to make a long story short, in a nutshell, I mean, let's say you have drugs that mechanism of action, let's say target inflammation, metabolic drugs, steatosis, and other more fibrosis. The issue is, for instance, with drugs like GLP-1 receptor agonists, you have weight loss and decrease in BMI. So if we use liver stiffness as a surrogate, does it mean that this is related to fibrosis improvement or just related to the loss of weight? This we don't really know, actually. It's not completely clear. Also, if you have a very powerful drug, does it make a difference in terms of liver stiffness of NIT? This we don't know. Just a parallel with viral load. If you have a 1 log decline, not sure you're seeing many things. If you have a 3 log decline, that makes a huge difference. And this we don't know.
Point well made.
So many questions, few answers.
Yeah. And then, of course, the question becomes, as we're dealing with regulators who need to be inherently conservative, I don't know if you caught the entire 2 days, but a whole session the first morning was dedicated to FDA cases. And part of it was, how do we make a decision? But all the decisions they talked about were things that had been controversial in the past. Alzheimer's, Makina, which they just withdrew recently, and then rosiglitazone, high profile cases, and the agency is conservative and cautious. So when we meet their data threshold, it may be very different than when we meet our own. Thank you so much for your time today, and I look forward to seeing you soon. Okay, thanks.
Thanks very much.
Bye-bye now. And finally, Veronica Miller. After Veronica, stay tuned for the episode 41 business report. So if I go by the day chronologically, this is our first interview of the day. I don't know yet whether it will be the first in the order that we place them, but Veronica Miller was good enough to join us before her day starts. Good morning, Veronica. How are you today?
Good morning, Roger. How are you? I'm fine.
I'm fantastic. And the ever-heroic Louise got off a plane from Perth, slept for a couple of hours, and is joining us for the interview. Louise, are you awake yet?
I'm awake, but whether I'm awake when we get to the last interview, we will wait to see.
And with that, why don't we just dive in? So, Veronica, fascinating couple of days, yeah?
It was intensive discussions, very good presentations, very good material presented. The organizers at the FDA did a terrific job of pulling it all together in the way they did. Now, of course, we have to then think about what was presented and what will the conclusions be that will be drawn.
So in the overall context of the organization and that statement, and I agree with you, I thought the presentations were fantastic and the organization of the event was was wonderful. What do you think might have worked better, if anything?
That they could have done better, really helping us understand how far they think we have come. In the end, I think there was a good summing up of the meeting. But these kinds of discussions, will just take an awful lot of additional discussions and in different settings to really drill down to the bottom of it. So a lot of was raised, for example, in that last panel about how we look at these non-invasive tests, right, in combination by themselves. And just because we look at them in combination doesn't mean it's necessarily immediately going to be a score or an algorithm. It's looking at individual patient level data versus population level data, etc. There's so much that still needs to be discussed about how they will actually be used. But I think it was just a good step forward in terms of having them as an audience to present to. And by them, I mean our colleagues at the FDA. They've got a tough job, They've got a tough job. And they reminded us at the beginning of the first day that things can go wrong. And they have to protect the public health, but they also should be promoting health. It's in the end an individual decision. And of course, within a division, they probably have a division level decision, but it's a decision of how much uncertainty is too much uncertainty, and how much uncertainty under what context are we willing to live with, and who takes the blame when that uncertainty turns out to be, we should have really been, much more cautious, because they talk about the Alzheimer's case, the McKenna case, the cardiovascular case, of course, and certainly where things have gone wrong, where things haven't panned out in the clinical endpoint study. But at the same time, in the Alzheimer's case, they pulled together data that convinced them, and it didn't really convince many other people outside of the agency. It's a difficult task to do this, and NASH is a complicated disease.
All true. And there are good reasons that many of us have chosen not to want to work in the government, even though we appreciate the importance of what they do and the value of it. I personally don't have the temperament for it, I have to confess. And when I was young, I really didn't have the temperament for it. With all that said, as you said, a good step forward. What were some of the things you think worked particularly well besides the quality of the presentations?
It was good to have, the presentations followed by the panel discussions. I don't know how many questions from the audience actually got to be asked and answered, because there was certainly enough just within each panel to discuss. There was clearly consensus on how ballooning as a diagnostic and prognostic and outcome marker just does not work. So hopefully that will be something that the consensus reaches them as well. And what I was also surprised at the end was how the statistician, Dr. Johnson, mentioned that, I think that was the clearest statement yet, about why are you trying to validate a surrogate with a surrogate? Why don't we just go right to the clinical outcome? People have just looked at the wording of the guidance in different ways. So I think that was really, really clarifying. But then how do we move forward to get to the next step? And what is the definition of reasonably likely? It's a definition that can't be put into a formula. It's really based on how do you look at the data and how convinced are you by the data? Different people will come up with different conclusions. And like I say, whether they do or they don't, they're always blamed if something goes wrong. So it's a tough job they have, and they deserve our support.
We agree. I've got a thought, and then I'm going to turn to Louise and see what she'd like to ask you about the overall event. I don't know if you listened to us back in our very first year. Most people couldn't find us. But right after the first OCA non-approval, we had on an FDA consultant who basically said that if you went to the cocktail parties, people would tell you that they would like to do away with surrogates entirely and just wait for clinical endpoints. Mm-hmm. Stephen Harrison's a big guy. I thought he was going to come through the wire, literally, and throttle this dude. I don't think I've ever heard him so exercised in my life on the idea that there might not be surrogates. Louise, you recall? Louise is smiling.
I wouldn't want to be throttled by Steve.
No, not even through a telephone, I got to tell you. But as I was listening to the questions about a surrogate of a surrogate yesterday, I hearkened back for the first time to the idea that there's probably a lot more sentiment behind that statement than we ever hear about on this podcast. So I'm glad that they noted that. Louise, you got a question for Veronica before we kind of move on next topic?
I did. And it was related to Laurelie. To echo what you said there, she challenged that way of thinking and for outcomes. outcomes. And I suppose that raised a question for me in the context. We focus very much on liver-related outcomes. We've had discussions on the podcast before that if we only keep it to liver-related outcomes, we're talking about a small proportion of all-cause mortality. But if we looked at liver health related outcomes, improvements to cardiovascular disease, improvements of diabetes metrics, things like that. Do we look, on listening to her and the feel of that last session, do we look to broaden the outcomes that we assess, or do you think we still need to keep very specific to only liver related outcomes because they take a long time to achieve?
Right. Well, that's a very good question, Louise. It will be interesting to see if the new definition or the new nomenclature, which is metabolic associated steatotic liver disease, will change any of that. So traditionally, and teaching medicine and all the work that's done in different agencies related to health, etc., it's always been very organ specific, right? So you treat the liver, you look for liver outcome. You treat the heart, you look for a cardiovascular outcome. And even though these are so interrelated, it behooves all of us to really look across these organ systems. And if the liver improves, does the cardiovascular system improve as well? I think is really, really important because of how they're all intertwined. So you have to look, in the end at what comes out of the guidance or what the individual sponsors, when they have their meetings with the FDA, what is decided. But generally speaking, I think the field is moving more towards recognizing that. But that topic wasn't even addressed. I didn't really see that being addressed over the last 2 days.
I thought it being addressed slightly in one of the presentations where they talked about the idea that from F0 through F3, the major causes of mortality are not liver. The statement was made, but right.
But
I think it kind of hung out there. It wasn't something that got a lot of attention.
The statement was made, but yeah.
I have one more question about surrogate of a surrogate before we go on. I think the logic behind Stephen and Mazen doing the NAIL NIT consortium was specifically to get away from surrogate of a surrogate, leapfrog biopsy, and go directly from surrogate to outcome. Did it strike you that what they're doing aligned with Dr. Johnson's statement, or was she off in a different direction even than they're going, do you think?
That's hard to say because like I said, I was a little bit surprised, with the clarity of her statement there. And I think I can't really speak to that in terms of whether she was, I mean, it seems like, they're going in the same direction. And, the NAIL NIT is one way to do it, but I think many others are already looking at that as well in terms of individual or combination of tests and how that impacts. But, we need good infrastructure. We need long-term studies to be able to look at that, of course. The comment that she did make was, the reason she was not convinced that some of these NITs predict clinical outcome is that so many of the studies were relating it to the biopsy.
Yep. In fact, I thought specifically that was the surrogate of a surrogate point, is if the biopsy is a surrogate, then there you go. So in understanding that this is a complicated process and that this was only the first stage, there's a metaphor about you got a lake, you drop a pebble in it, it makes a small ripple. You drop a boulder and it makes a much bigger ripple. How big a ripple do you anticipate this will make over time on how the agency looks at these issues?
Well, I think the pebble and the boulder is a good metaphor, but those are independent events. Somebody throws the little pebble and then somebody throws in the bubble. I see this more as a continuous progression where the little pebble that was dropped in has other pebbles that join it. I don't know how to explain the metaphor with the pebbles and the boulder, but...
Let me try it this way. A speedboat comes through the lake. Obviously, it will swamp anything but the biggest boulder, okay? On the other hand, a fishing boat comes through the lake. There may still be ripples from the stone, depending on the size of the stone.
Right. So I think it's a start that will initially maybe start with a little ripple, but that ripple will grow. It's not, that the ripple is there and then it disappears. The ripple will grow because the data that will be coming in. So, it's really the metaphor really is the data. Do we have a little data or do we have a whole bunch of data? And we're building on the data. So I see it more, maybe a better metaphor might be a snowball, right? That it starts small and then it gains size and power as it progresses. Because because I think once you start and people, untying the knot, right? Once you get the right place to start pulling things apart, then things become clear, things become more aligned, and we can move forward. But what is very, very clear to me, and this is why I always appreciate what Quentin says, is that we need to all pull in the same direction. We all need to kind of measure, report out things so that we understand if this NIT changes with this mechanism of action, but we've never measured it with another mechanism of action. How do we really understand? So we need to all measure the same thing, report the same thing, so we can really learn. And then collaborations like our placebo database at the forum, like the NIMBLE, which is continuing. Litmus has done heroic work. Unfortunately, their funding has stopped now, but it takes a lot of infrastructure and collaboration to get there. Just even a little bit more of an alignment will have a huge impact. Maybe not today, maybe not immediately, but you start aligning a little bit and suddenly you have a path forward.
It certainly conveys what the work the forum does and the way that you folks do it, that is the perspective that underlies it and makes, I think at the end of the day makes a ton of sense given the focus. Other than Dr. Johnson's comment about surrogate of a surrogate, what surprised you most, if anything?
Yeah, because I guess I know our colleagues at the FDA and I know the presenters, so there weren't really any other surprises. People said what I kind of expected them to say. I think maybe the biggest surprise is that the FDA actually held this meeting. And, these meetings take a long time to plan, and the FDA bureaucracy moves slowly and the administrative processes. So we know that the decision to have this meeting was made a long time ago. So I think I would really like to congratulate them on the job, the work that they did to put into this, and that it came out the way it did. So
the only surprise might be that there were no surprises, but that doesn't even sound like a surprise. It sounds like this is a fairly predictable event on that level.
But that's from my perspective, because I know, I'm sort of living this day in and day out.
Which is the only one we've got. I don't know, Louise, you have a question or thought about that? Was there anything that surprised you that's worthy of sharing with Veronica?
Yeah, there was something that surprised me. The lack of data on MRE for the majority of the outputs. And I think it was Dr. Circulating who presented that work, and he couldn't advocate for MRE in a number of areas, but also the lack of data that was available. So we talk about MRE being very prognostic in various areas, and we've talked about it on the podcast, but I wasn't aware that there was so much lack of data until that presentation. So that was my big surprise of the weekend. I echo Veronica's discussion there on the FDA calling the meeting, because for me, what it raises is, yes, biopsy is a surrogate, but I think Professor Banerjee also was very clear that surrogate that we use as gold standard comes with a 2.3 ratio of problems, including death. So we are choosing to use a surrogate that the FDA approved that has death as a related outcome. How many other clinical trials require such extreme outcomes to prove the outcome of your drug? I don't think there's very many. When we now have, and I think this was the strength of the panel that you chaired, we have as much evidence, if not more, to prove a panel of NITs that do not put patients at risk. And to me, that would be the concern, is that can we go forward with a life-threatening outcome required for clinical trials? Is that something I would put myself at? No. But we are asking patients to do that.
Right. And certainly if I was asked to give a liver biopsy, I would think about it very carefully, because I'm not very brave when it comes to having big needles sticking into me. Yes, that was a good point to have mentioned. I know we've often been very cavalier about a biopsy. I remember in the hepatitis C days where people with hemophilia were talking about their needs in a clinical trial and the need for a biopsy. And some people were very cavalier and say, oh, it's no problem. We can manage the risk. And, especially when you have somebody with hemophilia and you want to take a chunk of liver out of there. So I think that is a problem. I just wanted to say one thing, Louise, because I'm really a stickler for the language here. So the FDA has not approved the histology as an endpoint. The FDA is accepting it as a reasonably likely predictor. So the word approved, when we talk about regulatory agencies, sort of carries different weight. You're fully right, and I've put the challenge to people that if we could measure the volume of one liver biopsy, it's measurable. It's like a piece of pencil lead, right? And when we look at all the clinical trials and all the biopsies, how big a plate of chopped liver basically would we have, right? We're taking a lot of liver and looking at it, and this is the part also that I am really, myself personally and many people in the liver forum are pushing, that if we are going to take chunks of liver out of people, then let us analyze it as best as we can and get the most information out of there. So it might take a while to fully understand AI, ML, and how that applies to liver versus tumors, etc. But at least let's initiate that effort to really understand what are we looking at, something beyond the scale of 0 to 4. If we're still doing biopsies, let's at least learn more from them. It's something that we simply owe the patients. I don't have to list all the reasons why that is a problem, but that's always what happens, is sort of the first thing gets grandfathered in, and then everything else needs to be measured against that. And technology often advances so fast. We saw that, for example, with sequencing of genes. The old Sanger method was what was grandfathered in, and then we had deep sequencing, and then it was like, how do you prove that deep sequencing gives you the same result as a Sanger sequencing? So we're always going to be trying to take something new and understand it in the context of what we have already been using. And that's just somehow how this works. It's a challenge to keep up with technology from a regulatory viewpoint.
So listening to that last exchange, I had a thought that had not quite struck me before, which is that the patient advocates, who are the people who really should be talking about the 2.4% significant injury rate and the idea of death rates, have been a little too empathic around this issue and not data-driven enough. That if you look at the professional critiques or the academic critiques of biopsy, it's mostly about reliability and all that other stuff, which is really important. I mean, when we started this podcast, I wanted to describe it, and this is 4 years ago, I wanted to call this the Pyrite standard, and Stephen wouldn't let me do that. But I think it really is a Pyrite standard, and I think that's the fairest way to describe it. That said, the real issue is that if you put a drug in front of the FDA for a long-term chronic disease, limited levels of short-term mortality, with this level of risk in the testing method, I don't know that they would accept it. So I get your point about grandfathering, but I also suspect that the advocates haven't made a strong enough case on this yet.
That, I think, could very well be. And the data about that 2.3%, I have not looked at that. I can't speak to that specifically. I know it was mentioned. But certainly, I can imagine that, first of all, the unpleasantness of it, also the social burden. I mean, how much time do you have to take off work or not be babysitting or not, babysit your own kids? I mean, how much time do you have to take away from your daily life to just go through that procedure? That in itself is a burden, certainly the risk associated with it. So I can't really speak to the level of risk, but certainly it would be very nice to do away. It would have been nice also to have the patient voice in here then to make that point. But the decision was made to really focus on the data at this time. But I think, patients should have a voice to also say, this is not acceptable to us. If that's how they feel. I'm not telling patients what they should say. Just if that's how they feel, they should have the voice to say that.
We all talk to enough patients that we have a sense as to how that conversation would come out. I want to ask you about one specific point that struck me, and then I want to go to closing question. One of the few things that really made me wince, and this was in Dr. Sirlin's presentation, was his qualified but reasonably strong support of the idea that MEFib probably is a better test than MAST. And what it reminded me of actually was the London session in 2022 when Rohit presented those results, made a version of that claim, and then Mazen and Laurent kind of shot it out of the sky. I didn't think it was necessarily a good idea to air that piece of laundry yesterday if there were a way to avoid it. I don't know that there was. People don't collaborate on their work, and there are two schools of thought on this. But that was one place where it became clear to me that if I were the agency, I would throw up my hands and say, boy, I need a lot more data before I step into this.
Yeah, there were many, many instances of that, of where it's there, the data was presented, but I think most of the speakers also kept saying more data is needed, but this is, and I think Claude Serlin also made it very clear that, he would talk about what he knew as facts, or at least what he knew, and when he was expressing his own opinion. But I think overall the balance, amongst all the speakers came through.
Did you hear any other dynamics where if you were the regulator, you would walk away and go, okay, this is really unsettled, even by, not by regulator standards, but even by scientific standards? And I think regulator standards are tougher and probably have to be, but.
Yeah, maybe some of the discussion around, is it inflammation or is it fibrosis? Again, I thought just saying, well, in the end, it's the outcome that's associated with it, and inflammation and fibrosis are so intertwined again. There were a couple of instances like that where I thought, well, we're kind of getting lost in the weeds and are forgetting the thread of the conversation.
From your perspective, and I think your perspective is really unique because of the forum and the work you do and your mission, what impact do you see this having, to the degree that you're confident you can qualify it over, say, the next year and then over the longer term? Understanding that this is a ripple that will grow over time. Let's see if we can time the growth and the direction in which it's going to ripple a little bit.
Right. So again, to use the terminology, so the qualification process is this biomarker qualification program that is lengthy, arduous, intensive, etc., that many people are in. And the problem with that actually is that it's sort of one biomarker at a time and one context of use at a time. So that will follow the programs and the program's timelines. Where I would really like to see some change is in this area of what is reasonably likely to predict, which is a step before that, before we qualify something, and pulling together as a field to really get that clinical endpoint data through collaboration, et cetera, because no one can do that on their own, to come up with the clinical endpoints. The other area here where there's a lot of progress in the field generally is how we then bring real-world data and observational studies into the picture as well to help nail some of these things. And one of the speakers talked about hepatitis C, and based on epidemiologic data, the regulators said it's reasonably likely that the viral load would predict the clinical endpoint. It's so different with the virus, and it's the same division that the first surrogate in that way was HIV viral load. So it was sort of a natural progression from that. And I don't think anyone was surprised that the FDA came. But HCV viral load did not go through a biomarker qualification program. It was just accepted to be reasonably likely and then validated to actually predict through the studies that were done. The ripples that I see, or the little, snowball coming down, is really that kind of movement to what can we agree as a field, what is a reasonably likely predictor, diagnostic, predictor, prognostic, and especially the surrogate to predict clinical events. As we learn more and more about, the whole collagen, for example, as we learn more and more detail about what happens in the fibrotic process and really understand how that then could affect, long-term outcomes. And of course, the long-term safety of the drugs has to be documented as well, because fibrosis does play a major physiologic role. And all of us, if we didn't have fibrosis, we wouldn't have wound healing, for example. We need to look at, in the end, also at the long term, and also the strategy of how we use these drugs in combination. So a lot more room for discussion, but I think the really encouraging thing is that the agency has opened the door for this discussion. So let's look at it. They've opened the door very formally, because this was a very formal setup. It was coordinated and run and sponsored by the agency. Last I heard, a couple of days before, they said they had over 2,000 people registered, and I'm sure there were even more. It's a big open door. We can keep walking through it.
Louise, before we bid adieu to Veronica, do you have a question for her? Then I have a very, very quick closing question. I think that kind of was the closing question. So
no, I think she summed it up beautifully. Let's put a doorstop in the door from closing and just keep it going in the right direction.
Right. We all keep walking through the door, and we will get there together.
And pushing on the snowball, as it were, huh?
Yeah, it's the scary one, right? We don't want to have an avalanche come down on us, but...
No, and we don't want global warming to wipe the snowball away either. So, I mean, Veronica, thanks so much for your time today. It's been great of you to join us.
Thank you, Roger. Roger. It's always a pleasure, and have fun with the rest of them.
And now for the Season 4, Episode 41 Business Report.
Slava Ukraini, Heroyam Slava, glory to Ukraine, glory to heroes. I make it a point to refrain from commenting about U.S. politics or most things not related to Ukraine during this portion of the business report. But this week, I find it somewhere between frustrating and folly that one of the issues driving far-right resistance to moving any federal budget forward, thereby forcing a government shutdown, is that group wants to stop funding Ukraine war efforts. Crazy. As President Zelensky noted in his speech at the UN this week, Ukraine is asking us to give money in support of freedom and democracy while they pay with their blood. It seems the least we can do if we believe that democracy matters. I believe that democracy matters. Do you? Slava Ukraini.
September has been as big a news month as anticipated.
Paris clearly lived up to its billing, and the FDA NIT workshop feels like a pivotal moment in the progress of treating this disease. As I narrate this business section, the first day of the SLD Summit is going on in Prague, and I've already heard some fantastic talks. I anticipate more to come. You'll hear our episode on this next Wednesday evening.
Big news coming next week.
At the end of next week's episode, we'll have some major news about this podcast going forward. If you ever felt the desire to listen to this business report, next week you should be certain to do so.
No vault this week.
The 3 interviews in this episode cumulatively run well over an hour. So instead of sharing a vault this week, we will simply create excerpts, probably 2 excerpts from each interview over the weekend. That would be 6 conversations, with me sharing a few thoughts on each one, and vault will be back next week. That's it for now. I want to thank Jorn and Louise for joining us throughout the day yesterday, despite Louise's jet lag and Jorn's other commitments. I also want to thank Veronica, Laurent, and Naeem for some fascinating conversations. We record our SLD Summit recap next week, along with news about the future of the podcast. Please join us starting at 10 p.m. Eastern U.S. time Wednesday night to hear it. And until then, stay safe, surf on, and we'll see you next week on the podcast.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.